So hello there, welcome to this video. My name is Yuri Chemale. I'm a senior TA applicant, and today I'll be discussing with you about the principles and practice of vaccinology. So just a brief introduction, vaccinology can be said that it's the study and development of biological products that can safely induce an immune response against a communicable disease. And it's a very clever way that we have designed to fight these communicable diseases because we are utilizing our latest technology by producing these very clever vaccine antigens and then presenting them to our immune system that has been evolving for the past millennia in order to produce this immunity and reduce either infections or hospitalizations or worst case scenarios due to the production of antibodies either to the memory cells or the cellular pathway. And this is particularly important because infectious diseases have been a major cause of morbidity and mortality throughout human history. And of course, if you take a glimpse at just from the year 2000 from now, we see that they're still important, being like 3 out of the 10 leading causes of death globally being from communicable diseases, and particularly true for lower income countries where there's still a high burden with 6 out of 10 of the leading causes being from communicable diseases. But it's also important to take a look at it through the eyes of history, because if we look at from the 18th century, 19th century, and 20th century, we see that they played a much larger role in the deaths of human beings worldwide. And this reduction is certainly due to many factors, primarily being from the development of new treatments, such as the discovery of penicillin and the production of new antibiotics, as well as improved sanitation, but also through the development of vaccines. And an important fact is that the WHO estimates that 2 to 3 million lives are saved each year due to the immunization coverage that we have nowadays. And of course, this is not counting the lives saved during the pandemic and the impact that this had on this data as well. But if you take a look at our countries that are highly immunized, such as Australia, where we have that from almost 2011 to now, a high coverage on children 5 years old or younger, reaching almost the 95% aspirational targets that they have set themselves. We see that some diseases that had severely impacted the lives of our forefathers, such as polio, pertussis, diphtheria, tetanus, measles, mumps, have dropped down to nearly a negligible number of cases per 100,000, and certainly a negligible number of deaths and morbidity as well. So this is just to highlight the importance that vaccines have had throughout the human history in controlling these communicable diseases. So now I'd just like to talk with you about the vaccine development part. So to start, the vaccine development has some particularities that we have to be mindful about. I mean, it's not the same as just a regular randomized controlled trial. First of all, it's because the diseases that vaccine are trying to treat, they have varying incidences during different time periods. I mean, just look at this graph. We can see here that the notifications per 100,000 population of diphtheria across this time frame, we see that if we had started a trial around 1922, we would have reached the peak of the incidence of diphtheria, and this would produce drastically different at least results from if we take a look at 1926, where there is a steep drop off, and even more so later on, more recently. The same is true for pertussis, where we have this varying incidence rate, and of course, for the COVID cases as well. As we can see here that there was a peak, this data from South Africa, there was a peak from around June 2020 to September 2020, and then there was a peak again on the late November to early to early April, and so forth. And these differences is something that we have to account for and plan for when we're designing a randomized control trial. And another key impact that communicable diseases have on vaccine development is that vaccine development can induce what is called the herd immunity, which means that if we have enough people of a community, of a group, immunized, not necessarily vaccinated, but immunized against this disease, it will stop the spread from people that are infected to people that are susceptible to this disease. And of course, this is something we do not observe in other types of treatment. I mean, if I'm taking statin, for example, I won't prevent the cholesterol development of my wife, Gabriella, or for other people near me. Meanwhile, vaccine and immunization has this can have this impact as well, this indirect protection. Well, but aside from that, the development of vaccines, they follow a very a similar path to other major substances. We need to start with a phase 1 trials where we are trying to assess whether the substance is safe and we can actively use in humans. And we follow through that with a phase 2 trial where we're looking for signs of efficacy with a small sample size. Nowadays, most of the trials are using this phase 2/3 designs, where instead of just doing a phase 2 and analyzing the results at the end and publishing it, and afterwards proceeding with a phase 3, they develop this a bit more complex designs where it's included in then a first stop, just look for efficacy, and then utilizing these data, we can move on to a larger trial and see if it's generalizable. Just a brief point on vaccine efficacy. When we're trying to test this intervention, we're looking whether there is a reduction in the incidence of diseases in the vaccinated group versus the unvaccinated group. And this is something that it's a gold standard in practice, which is to use what is this formula here, where we look at the incidence in vaccinated group and compare it with the incidence in the unvaccinated group, and produces this, which is basically just the relative risk reduction. And I always wondered, why are we using the relative risk reduction? I mean, why not use the absolute risk reduction, which is something that has been has been more used or been more stimulated to be used in other diseases. And this can come through basically 3 facts. So the first one is that the relative risk reduction is indifferent to the transmission setting. I mean, imagine if we're designing a trial again during these peaks here on these diseases, we would have a drastically larger incidence rate in both groups, and this could certainly provide a drastically larger absolute risk reduction, right? But it won't be the same as if we designed a trial on this lower end, such as in 1932. So the relative risk reduction is more robust to changes in transmission settings. And as there's a high variability among this, the transmission rate during a pandemic or epidemic or endemic, it's sometimes better to have an understanding of what is this relative risk reduction. Moreover, another benefit of the relative risk reduction is that it's usually roughly constant across different subgroups. I mean, if we are trying out a vaccine again in a population, and we can separate this population into high risk group and low risk group, we see that if in the high risk group we have the control rate, which is about 40%, and the vaccine can provide, for example, a 25% reduction on this. Into this, there will be a 25% reduction, which will come to 30%. But and whilst the absolute risk reduction will be 10% between those groups. But if we look at low risk groups such as young people, healthy with no signs of comorbidities, the relative risk reduction will still be 25%, but the absolute risk reduction will be just 2.5% if the base rate is 10%. And lastly, the absolute risk reduction will always appear low in low event rates, and in randomized control trials, at least in the moment, we do not expect to actively expose our participants to this sort of disease. So this is also something that we always have to have in mind. I mean, if you look at the trial data from Pfizer, CoronaVac, and Moderna, we see that their event rate was quite low in almost all of them. You could say that in CoronaVac it was a bit high given their choice of population. But in the Pfizer trial, if we just look at the absolute risk reduction, it's a measly 0.71%, and this would provide a number needed to treat of 141. Whilst on the CoronaVac trial, there was an absolute rate reduction of 1.8%, which is certainly more significant if we look just at this data. But again, this is just a glimpse, and if we account for the transmission settings on both trials, where Pfizer was using community data and CoronaVac was using healthcare workers, it's certainly something that we can't compare one to the other. And the benefit of using the relative risk reduction is that we do have this number that we know that it's probably going to reduce about this disease if you are vaccinated or not. So now, after you develop a trial, you see that it works, it has a good vaccine efficiency, we can start looking at the vaccine effectiveness, which is how does this vaccine work on the real world, which is much more important, right? So the first step that we can do that is we can have three ways of doing that. We can either do screening data, we can look at case controls or cohorts, which are both types of observational studies. And the screening data is much more it's much faster and easier to do, where you basically look at the percentages of cases vaccinated and the percentage of the population vaccinated. And this can give us a rough estimate of how much the vaccine is working, right? This is the CDC, Center for Disease Control, has been doing so ever since vaccines have been updated, where they are looking at the rates of vaccination, and they are saying that, oh, in October, unvaccinated people had a 5% more risk of developing COVID, and a 14+ risk of dying from COVID. And another way is doing these observational trials, which can be a case control or can be a cohort. And if the disease is in a low prevalence rate, we can say that the vaccine efficiency is 1 minus the odds ratio. We can look at that in this trial now that has been published by Zhang et al., which is actually a real-world effectiveness of COVID-19, they're doing a meta-analysis of over 50 articles using 39 cohort studies, 12 case control studies, and data from 14 countries. And we see here that this trial, they have a very good, how can I say, high quality study. They have high quality studies in this meta-analysis. They're using to assess that through the Newcastle-Ottawa scale, which ranges from 5 to 8. And we see here that in this trial, those trials over here are from the Pfizer, those are from Moderna, and those are from CoronaVac, and there are about 27 studies that has been published throughout multiple countries on the Pfizer BioNTech. And we see that this real world data stating that the Pfizer BioNTech is helping, there's a vaccine efficacy of 91.2%, ranging from 87.9 to 94.5, and these results are statistically significant. And if we go back to the trial to the results from the trial, we see that this confidence interval is within the limits that were set out during these early trials. And of course, the Moderna, though there is no statistical significance, they probably given the low number of events, there is there's a very high vaccine efficiency, and the CoronaVac, there's a lower vaccine efficiency than the other ones. So just to summarize what we have been discussing so far, the communicable diseases, they poses a high burden throughout human history, though there is a diminishing burden of it in the recent days. It still is very important, especially in lower income countries. We now, well, we know that vaccine saves millions of lives worldwide, and that is an annual, in an annual basis. And I do not know of any other intervention that has this impact, this consistent impact throughout human history. And lastly, we briefly looked at the vaccine development, looking at this how varying transmission rates can impact the results of a study, and how vaccines can provide either the direct and indirect protection. And lastly, how do we assess the vaccine efficacy, and how, after we have done that, we need to keep on looking for this vaccine effectiveness, right? So this is just a reference that I've used, and I'd like to thank you all for staying here with me, and I hope you all learned something new today. And please, if you have any comments, any doubts, or if I missed anything, please just comment here on River, and we will keep on this brilliant discussion. So thank you all, PPCR, and I hope you have a marvelous year here in 2022. Cheers.